Okay, hopefully this is working. So I just wanted to go through the exam one review really quickly. I won't go too much into depth, but I did want to go over it with y'all before the exam. And do remember that we still have class on Monday, so some of the stuff you may not know yet, and we will be talking about it on Monday. But yeah, let's get into it. So a gene is a segment of DNA that contains info to produce a function. Okay, and a trait is the physical characteristic of an organism. So, okay, an allele is a different version of the gene, so an alternate form of a gene. Okay, genome is a collection of all genes. Proteome is a collection of all proteins in a cell type. And just remember, some cells are specialized, so they have different proteins in them at different concentrations. That's why it's a little different. Okay. Homo Homologs are homologous chromosomes. These are not identical chromosomes. You get one from dad and one from mom. Okay, and sister chromatids are 100% identical. So identical. And this happens in mitosis and meiosis. Two. Okay, natural selection is when the environment selects traits that lead to greater reproductive success. Oops. Okay. Morphs. So this is a form of phenotype that varies within a population. Phenotype that varies. So this would be, I don't know if y'all got this example in class, but it would be like frogs and they're the same species, but they look really, really different. Genetic variation is the genetics differences among a species based on the changes in the DNA. Now, Mendel's law of segregation. So this is separating homologs, and if you remember, it's in anaphase one and anaphase one. Okay. Now the law of independent assortment. This is the random alignment of homologs, and this happens at metaphase one in metaphase one. Okay. So the synaptimal complex links together homologs and meiosis one. Me- one. Okay, the chiasma is a structure formed when chromatids cross over. So, okay, and bivalents are just homologs. And if you remember up here, homologs are not identical. So you get one from mom, one from dad. Okay, dosage compensation is when the levels of expressions of genes on the sex chromosome is inactivated. Levels of expression of genes, and this is specifically on the sex chromosomes. Okay, so an example of this is our X chromosomes are huge, right? So we have lower expression of our X chromosomes versus our Y chromosomes are, let me try to do that better. They're tiny. So we have higher levels of expression in in the in these sex chromosomes. So go back to sex-linked inheritance. So sex-linked is X-linked. Sex-linked and is linked to the X chromosome. Chromosome. Okay, endosymbiosis is a really relationship in which um, an organism lives inside its host. So in our case, it's chloroplast, and mitochondria. And this is both beneficial to the host and the organism that is living inside the host, which is mitochondria in our case. Okay, over dominance. So this is the heterozygote has the advantage. So an example of this would be sickle cell. Okay, incomplete dominance. This is when there is an intermediate phenotype. So that would be we have a red gene and white is not going to show up. So, and then we have a yellow gene, these two mate, and then we get orange. So, gene interaction. The presence of two genes influence the outcome of a trait. I'm gonna move this to the side. So an example of this would be like in skers, if you have, I'm not sure if it's skers, but I want to say it is. So basically, if you have two recessive, it blocks out a dominant. That would be an example of gene interaction. Okay. Now we have lethal alleles, which kind of in the name, an allele that can be deadly. Epistasis is when one gene masks the presence of another. Pleiotropy is when one gene has multiple effects. Incomplete penetrance is when a dominant phenotype is no, a dominant genotype is carried but is not expressed. So this would be our an example of this would be polydactyly. Polydact. I think that's how you spell it. I'm not sure. Um, variable express expressivity is a spectrum of phenotypic expression. Okay, cytoplasmic inheritance would be the inheritance of extranuclear. Wait, yes. So this would be mitochondria or chloroplast in plants. So heteroplasmy is a condition of a cell in a particular organelle. Okay. Gene imprinting is when a single a change in a single gene or chromosome during gamete formation. So an example of this would be methylation, which is just turning something off. Okay, paternal leakage is when a dad provides the mitochondria. Okay, paternal effect is when the genotype of the mother equals the phenotype of the offspring. So an example of this is the snails. So if the mother's genotype is dextral, then the phenotype of the offspring will also be dextral. Okay, now for the conceptual part, what are the levels of genetics? So molecular, cellular, organismal, and population. So population would be something like natural selection, organismal would be something like phenotype, cellular would be cell division, and molecular, I'm just gonna put it up here, would be protein synthesis. Okay, what are environmental factors and how do they affect your gene expression? So, um, environmental factors can express the expression of your genes through the determination of sex in certain species. So I'm just going to write sex determination. And then we have PKU, which I think is the I think that's when you can't eat a certain chemical and you're screened for it as a baby when you're born to see if you have that disease. And if you do eat something that that gene encodes for, then you'll get really sick. Another factor would be like the fox changing coat. Okay, what are the biggest differences between meiosis one, meiosis two, and mitosis? So, meiosis one, you're going from diploid to haploid. Okay, and meiosis two and mitosis. Well, okay, meiosis, let's do meiosis two. You're going from haploid to haploid. And then in mitosis, you're going from diploid to diploid. Okay, what is the central dogma and its products? So Central dogma is DNA to RNA to protein. Here we have transcription. And here we have translation. And something that helps me remember which comes first is I was told you have to write something down before you can translate it. So you have to transcript it before you can translate it. So saying you have to write something before you can translate it. What are the Mendelian ratios for a monohybrid cross and a dihybrid cross? So monohybrid is um is one gene. And remember, we have phenotypic and genotypic ratios. So I'm gonna do genotypic first. Genotypically, it's 1 to 2 to 1. So phenotypic is gonna be 3 to 1. And then really all you need to know for a dihybrid cross, see, I'll try to fit it over here. Oops. Hope it's still recording. Dihybrid. It would be 2 genes, and they are heterozygotes for the gene. And that would lead to, and this is a phenotype of 9, 3, 3, 1. So phenotypically, that's the ratio. Okay. What is a pedigree and what does it look like for a dominant and recessive gene disease? Sorry. So for a dominant disease, there are no carriers and it's in every generation. And then for recessive, you have carriers and it kind of skips around because you have to have two, um, well, you have to have a homozygous recessive to have the disease. So it skips. And this has carriers. Okay, so what are the stages of the cell cycle and what happens during these stages? Sorry, it's my cat. Um, G1, growth. Then you go to S. synthesis, and then you go to G2 and say growth again. Also have some checkpoints. Um, and that's interphase, and then we go on to mitosis, and you have prophase, prometaphase, I don't think we're gonna have room for this, so I'm just gonna write M for metaphase, then you go to anaphase, telophase, and then cytokinesis. So, um, one thing that I kind of want to go over is like what happens during these stages, but using kind of the number of chromosomes in each stage. So let's say we have 46 chromosomes. Okay. And then in the S phase, remember that we're synthesizing the DNA. We're making copies, right? So here we would have 92. And then we would keep on having 92 because we're just growing. This is when the nuclear and prophase, the nuclear envelope is disassociating. The spindle begins to form. Prometaphase is when they attach to the chromosomes, but you still have 92. Metaphase, they line up on the plates. You would still have 92. And anaphase is when they actually get pulled apart. So you separate into back to 46 and 46. And for the rest of these, you're going to have 46 and 46 to give us back the same number that we started with in mitosis. Okay. And coming to this next question, it's important because I actually did get this wrong during study sessions. So I do apologize for that. So I want to clear it up. You only don't count the X's. Well, the only chromosome that gets condensed is an X chromosome. So this individual would have two, B would only have one, because again, we're only taking out the X chromosomes. This one would have zero. This one would also have zero because it's only X. So just X chromosomes get condensed. I spelled that wrong. Okay. And they would have one. Okay, what is the difference between haploid and diploid, and where do we find them in the body? So haploid is N, and diploid is 2N. And haploid would be the gametes, sex cells. Diploid would be our somatic cells. So basically every other cell but the sex cells. Okay. Hippos have 36 chromosomes in their somatic cells. How many chromosomes does a gamete of a hippo contain? So going back to kind of what we just talked about, if they have 36 chromosomes in their somatic cells, they have half of that in the gamete. So the answer to this would be 18. Okay. A deep golden is dominant to a pale golden. You cross heterozygous flower with a recessive flower, predict the genotypes and the phenotypes of the offspring. So you have a heterozygote and a recessive. Okay, so it looks like these two are dominant and will be the deep golden color, and these will be the pale. I'll just do the red. So we're about half and half. And that is also reflective of their genotypes. We have big G, little g, and small g, small g. So D is our answer. Okay, color blindness is an X-linked recessive condition. An unaffected mother has two children, including a daughter who has color blindness. What is the probability that the brother of the girl will be colorblind? Okay, so we have the mother up here and the father right here. We know it's X-linked, so he has it. I'll just do A. And the mother is unaffected, but her daughter has color blindness, and it's a recessive condition, so she has to be a carrier. So put it on this one. Okay, and we're looking for the probability of the brother of a brother of the girl who is colorblind. So we are looking at the boys right here. And just by looking at it, we can tell that we are going to have 50-50 because this one has it and that one doesn't. So our answer is C. So in some plants, blue flower color is dominant to yellow flower color. However, heterozygous plants have a green color. If a green flowered plant is put in a test cross, what will be the phenotypic ratio of their offspring? So we want to remember that a test cross is recessive. So when you're doing a test cross, you're crossing it with the recessive. So here we will have I'll just use a heterozygous plant. And we have, because we have a test cross, we know it's going to be tested with a recessive. So, oops. Okay. And we know heterozygous plants have a green color. So this is a heterozygote, this is a heterozygote. But the other two are recessive, so they will be yellow. And this is asking for the phenotypic ratio, so it's going to be half green, half yellow. Okay. Scurs are on cows are said to be a sex influence. If P is dominant in males and A is dominant in females, what would the ratio be for a cross between those genotypes? So first we have a present, well, we have two heterozygotes. So just gonna write them like this. Okay. So we want to remember that. Let's look at the girls first. A is dominant in females. So this would be dominant, this would be dominant, this would be dominant. So if that's dominant, then that would be three, 3 out of 4 females would have skirts. And now if we do males and that P is dominant, we would have this one, this one, and this one. Okay, so all females with A will have skirts. That is true because, I mean, because here, here, and here, all those females will have skirts. So all the females with an A will have skirts. So that, no, because A is absent, P is present. So all males with P will have skirts. Yes, because P is present, P, all the males will have skirts. That's true. So all females with recessive genes will have skirts. All females. Yes, because here is a recessive gene in females. Because A is dominant in females here. So all females with recessive genes will have skirts. So this is true because P is recessive in females and P is present for skirts. So the answer is both B and C. Okay. A mother who has type A blood type and a father who has AB, what is the probability that one of their offspring will have type O blood? So if we remember the blood types, type O is the two little I's. And just by looking at their blood types, we don't have two little I's to match. You could do the cross, but just by looking at it, there's no way that you could get a double I. So the answer is 0. Okay. We call that you have that in pea plants, purple is dominant to white. white is recessive. The two heterozygous plants are crossed. What is the probability that their first six offspring will have purple flowers? So two heterozygotes. I'll do big P, little p. Okay. So out of these, this will be purple. So we know that the probability for purple over white is going to be 3 out of 4. So the probability that their first six offspring will have purple flowers is you would just 3 out of 4, 3 out of 4. You just do that 6 times. And that would be your probability. If you do the math, it comes to 18%. Okay. Before the S phase, a chromosome has, I think that's a typo. Anyways, has 46 chromosomes. How many chromosomes are there on the metaphyseal plate? So this is something that I was going over earlier. So in the S phase, well, before the S phase, we had 46, right? So 46 we synthesized here. And now what would we have on the, well, we got a couple stages to go to, but what would be on the metaphyseal plate? So the answer to that is 92, because it would be double the 46, because here during synthesis you doubled them. Okay. A mother snail is sinistral and her mate is dextral. What will their kids' genotype be? So just by looking at their genotypes, we know all their kids will be heterozygotes. So all her kids will be DD. Well, big D, little d. And then a child from the previous part is said to be, will it be sinistral or dextral? So the answer to this is, has to do with the maternal effect that we talked about up here, right here. So when the genotype of the mother is the phenotype of the offspring. So the genotype of the mother is sinistral. So that means the phenotype of the kid will be sinistral. Because the genotype of the mother is sinistral, so the child will be sinistral. Epistasis is when one gene masks another. An example of epistasis in mice, you have the dominant and then the recessive. However, a separate gene necessary to produce a pigment. A mouse with a recessive C allele at this locus is unable to produce the pigment and is albino, regardless of the allele present. A cross between those two genotypes, what would be the phenotypic ratio? So we can tell here that we have two heterozygotes that are being crossed, and this is um, a dihybrid cross. So if we look back to here, number 5, we can see dihybrid cross heterozygous parents will produce a 9331 phenotype. So that will be our answer. 9331. Okay. You don't know if a pea plant has green or yellow seeds, so to find out, you cross the plant with green seeds. The result is a mixture of yellow and green seeds. What would be the genotype of the unknown plant be? Green is recessive and yellow is dominant. So I'm just going to use A's again. So we know the plant is crossed with a green seed, and green is recessive. So we know we have homozygous recessive. So we have little a, little a. And it says the result of the cross was a mixture of yellow and green seeds. So if we had, let's say we had a homozygous dominant, right? But if we did that, all the kids would be heterozygotes, and they would all be yellow. But that's not what happened when you cross them. When you cross them, you got yellow and green. So that means, oops, that you must have had a heterozygote, because that's the only way you would actually get. So yellow is dominant. So these would be yellow and these would be green. If it was a homozygous recessive, they would all be yellow. If it was a homozygous dominant, then they would all be green. But in the question, it says we got a mixture of green and yellow. So that means we have a heterozygote. Okay. That's all I have on the review. I do have office hours on Monday, and I don't think I'll be having a session on Tuesday just because the test is on Wednesday. I might just be there if you want to go over the review. If you can't come on office hours, I'll probably just hang out during the study session. We won't be going over anything. But if you have any questions or want to go over anything, just feel free to reach out, and good luck on your exam.